on DNA replication. We had gone through the whole process of DNA replication, focusing on eukaryotes, because of course we're talking about eukaryotes with cancer biology. I want to talk about the end replication problem. This is a problem that only exists for eukaryotes. It doesn't exist for prokaryotes. That's because if you remember that DNA polymerases always make DNA in the, which orientation? 5 to 3. They always make DNA in that one orientation. That means the very end of each chromosome won't be replicated on one side. Think about leading and lagging. Right? What will happen here, they're showing you from your origin of replication, you have a leading side, a lagging side. Right? The leading, you'll get full copying all the way to the end. But on the lagging, because you'll have those Okazaki fragments, those individual fragments, the RNA has to be digested out and then filled in with new DNA. But remember, polymerases can't attach to nothing, so when they fill in with new DNA, they actually grab onto this fragment to fill in that space. So what that means is on the lagging side, you'll have a little area where there's RNA left over. That RNA gets digested out, and there's no way a polymerase can fix that end. So what happens in a linear chromosome is every single time the chromosome goes through replication, it gets a little bit shorter. It's true for all linear chromosomes. Every single time you wind up with this little gap in that 5' end of newly synthesized DNA.
Is this what's considered a telomere?
Yeah, this is what's going to fix the problem, is the presence of telomeres. So it exists because of the difficulties of DNA polymerase has to attach to DNA to start its replication process, and the lagging strand. It always happens on that lagging strand side.
This is only true in somatic cells, though, right? Not in.
I'm going to get there. It is a problem everywhere, but there's a way to fix it in germline cells. So the problem here is fixed partially through the presence of telomeres. Telomeres are repetitive nucleotide sequences found at the end of eukaryotic chromosomes. There's usually 100 to 1,500 copies of a certain sequence. In humans, the telomere sequence is TTAGGG. That's the sequence we see repeated in humans. It's at the end of every chromosome. Every linear chromosome has this telomere sequence repeated many, many times, hundreds of times at the end. Now, what that means is, as the chromosome, I should back up and say, it doesn't code for anything. This is non-coding DNA. Just this telomere sequence that's repeated at the end. It does not encode any gene or anything like that. It's just sort of end DNA, creating this end to the chromosome. Now what happens is when this chromosome goes through replication, it will get a little bit shorter. It will with every round of replication, it will get a little bit shorter. But it's not a problem because you're kind of eating up some of that telomere, and the telomere encodes nothing. And you have hundreds of sequences there of that telomere repeated. So you have this kind of space at the end that can get shortened with each round of replication. It's not going to hurt the chromosome. It's not going to hurt any gene. That's present on all of our chromosomes.
Is it just on the lagging strand?
No, it's actually seen on both. It is seen on both, yeah. Because what will eventually happen is both strands will get shorter, because as replication happens, the other side will get shorter as well. It won't be able to copy. So they will both end up shorter. So the ends of all eukaryotic chromosomes are marked by these telomere sequences, this non-coding DNA that allows shortening without damage to the chromosome. Now, of course, this isn't a problem for most cells because most cells will only go through a certain number of cell divisions, but it does become a problem in other cells. And of course, the cells we're worried about are things that get passed on to the next generation. We're worried about eggs, we're worried about sperm, we're worried about those cells that go through a significant amount of DNA replication. They have to preserve their telomere sequence. So there is an enzyme that will address this problem, telomerase. Telomerase is an enzyme made of, it's an enzyme that has protein and also an RNA component to it. So it's both RNA and protein together. turns out that the RNA portion of telomerase is complementary to the telomere sequence. The RNA portion of that enzyme is complementary to the telomere sequence. So here's how it works. It's going to elongate out our telomere by grabbing on, here our telomerase enzyme is attaching to the overhang side, the 3' overhang. The chromosome has gone through cell division. You have this overhang on the 3' side. Telomerase will come in and attach to the 3' overhang. It lines up that RNA sequence, which extends out the end, basically. It's sort of extending out the end of the chromosome. That's going to allow polymerases to add on additional nucleotides. It's adding on according to the base pairs that it reads in that telomerase RNA. So it extends out the 3' end. Lest you think, wait a second, we're only making one side longer, not the other. Well, what happens then is you have a longer 3' end, which means you get another Okazaki fragment, which means you get extension on both sides. So telomerase extends the 3' end, and regular DNA replication will extend the other side during the regular DNA replication. So telomerase is an enzyme that extends out the telomeres and allows you to retain the telomere sequence, especially important in germline cells. That's where you see it. So it's active in germ cells, not expressed in most somatic cells. We're going to see in cancer cells, commonly telomerase is reactivated. Right? Because, of course, all cells have the same DNA. What makes one cell different from another is expression. Right? Think that through. All cells of our body have the same DNA sequence. What makes a skin cell different from a kidney cell is which genes are being expressed. They all have the same DNA sequence. Kidney cells only make kidney proteins. Skin cells only make skin proteins. So what's happening here is all cells have the gene for telomerase, but only germline cells are supposed to be using it. Cancer cells find a way to turn telomerase activity back on. That's how they can go through many rounds of cell division and retain the chromosome telomeres.
I read in the article that they're trying to extend telomeres because they believe that it's
aging,
like a fountain of youth.
Right. Yeah, that has been proposed. I don't think they have much evidence to back it up, but that has been proposed. In fact, one of the ideas behind that restrictive diet, you know how if you eat low calories, lifespan extends. We know that's true in mice and rats. Don't have a lot of data on humans yet. It takes a long time to look at lifespan. But one of the ideas is that you're going through less rounds of cell division because your nutrition is low, and therefore your telomeres aren't shortening as quickly. I don't have a lot of evidence to back it up, but this has been associated with aging, and possibly this is related. All right, so telomerase, active in germ cells and also activated in cancer cells. Now, an interesting thing that happens also with the telomeres, telomeres will also help to block degradation of DNA. What happens here is that overhang side, the 3' overhang, will actually turn back, and through base pair rules, it will bind to the another part of the telomere. Let me see that more clearly. Because it's the same sequence over and over and over again, the 3' overhang will actually turn around and bind inside of the telomere to create kind of a loop structure on the end of all eukaryotic chromosomes. The value of that is that it prevents degradation. The end of the chromosome is less likely to be degraded because you have this looped around structure. There's also some proteins that will bind to hold that loop structure in place. It's a way of preventing degradation of the telomeres too fast.
So the telomere binds into what part of the...
Inside of the telomere itself. So this is all telomere at the end here. Since it's a repeated sequence, part of this can come around and bind in here through base pair rules. Because it's the same repeated sequence over and over again, so it can come back and bind inside of there. And it is beneficial to do that because you don't get as much degradation at the end of the chromosome if it's in this loop structure as opposed to out in the cool aqueous world. All right, so The last kind of part of DNA to talk about here is packaging. DNA packaging, especially in a eukaryotic cell, is a huge issue because the eukaryotic chromosome is extremely long, and huge quantities of DNA must be packaged into very small spaces. If we were to take the DNA from one human cell and extend it out, it's about 6 feet of DNA that's crunched into the nucleus of one cell. So this is an incredible packaging ability that happens. It's true in both prokes and eukes, but especially eukes, because our chromosome has so much more DNA in it. So how does that packaging occur? There's several steps, but the real major players here in packaging of eukaryotic DNA are proteins called histones. Histones are proteins that have a lot of positively charged amino acids. Remember, amino acids can have various different qualities. They can be hydrophobic, hydrophilic, positively charged, negatively charged. Histones have a lot of positively charged amino acids. Why is that important? DNA phosphate groups are negatively charged. So what's happening is the negative charge on DNA, because of all those phosphate groups, is attracted to the histone protein. So it coils around the histone proteins in a very tight association. Sort of like a spindle, like if you think about thread on a spool, that's kind of the idea. The histone proteins make this spool, the DNA wraps around it sort of like thread around the spool there. So DNA wraps around this group of histone proteins. There are a lot of them in the group. Don't memorize the different numbers. I just want you to recognize that histones are packaging proteins for DNA. DNA wraps around them, and you'll have a number of different histones that come along the chromosome. So here you can see a group of histones, the DNA is wrapped around, about 2.3 turns is how many times it will go around, and then you'll have a little piece of DNA linking it to the next group. You wind up with a structure that looks like beads on a string, and these are groups of histone proteins where the DNA is wrapped around them, creating the first level of organization.
I'm just confused by the second statement. You say it's positively charged, but it's basic, as in pH, or basic as in basic amino acid?
Well, it's basic amino acid, not as in pH though. pHs would be, well, basic amino acids do have a positive charge to them. So yeah, I guess that's that is true. Basic amino acids do have
a negative charge, have an excess of electrons.
Say that again.
Basic would have an excess of electrons. That's why.
Basic amino acids have a positive charge. Am I remembering that wrong? No, I'm not talking about pH-wise. We're talking about, it's a description for amino acids. You can say it's positively charged or you can call it basic. It's the same description.
So basic is referring to its structure?
Is referring to the fact that that has a positive charge in its structure. Not the liquid around it. That's what pH would be referring to. It's talking about the actual structure of the amino acid has a positive charge to it. No, it doesn't mean common or basic. It means basic means positively charged. When you describe amino acids, you can describe them as being basic or acidic. Basic means that they have a positive charge to their structure. It's the same idea. So, positive charge that the negative DNA will attach to it via bonding. The structure of a histone with DNA wrapped around it is called a nucleosome. Right, so it's called a nucleosome once you had DNA wrapped around your histone proteins. You wind up with several of those together. They did an interesting experiment to show that these proteins show up about 200 base pairs along the DNA. What they did is they took chromatin. Chromatin is DNA with the proteins associated, so it's DNA with its histones attached to it. They took that chromatin and they digested it with a nuclease. Nuclease degrades DNA, but it will only degrade DNA that is accessible to it. In other words, it can only degrade this DNA. It cannot degrade DNA that's actually wrapped around the histone. So it only has access to this linker DNA. So the enzyme is used to digest available DNA. Right? And then you go through and you run your fragments on a gel, and what they see is repeated 200 base pair ladder. So you see DNA at 200 base pairs, 400, 600, 800, indicating that those histone proteins show up every 200 base pairs along the DNA. That's how common they are in the DNA structure. So interesting experiment to show it's actually a very predictable structure every 200 base pairs. From that level of organization, those nucleosomes are attached through another histone protein that packs them even closer. It will take these nucleosomes, and an additional protein actually pulls the nucleosomes together, forming another tight fragment here. So here we have our nucleosomes. They're all held together into this chromatin fiber. If you look at the structure of this, it's amazing. All of the DNA wrapped around these individual histones, and then the histones associated together to form a really tight structure of DNA and protein together. This is the 30 nanometer chromatin fiber. It's just the next level of organization, from DNA to these nucleosomes and into this fiber of all of the nucleosomes attached together.
How do they bond? How does the DNA bond to the histones?
It's just through interactions between the positively charged amino acids and the negatively charged DNA.
Would DNA methylation affect the every 200 base pair?
DNA methylation won't affect the histone structure or the binding to the histones. What it will affect is what we're going to see in a moment, which part is transcriptionally active and which part is not. So some will be really tightly packaged if it's not transcriptionally active, and some will be looser. Methylation affects that portion.
Okay, but you won't be able to see it
You won't be able to see it at this level. All right, the next level, these chromatin fibers that so far it's all histones and DNA, are then attached to a scaffolding protein. This is not a histone, it's another protein. You take this 30 nanometer chromatin and you loop it, and these loops actually are less regular. It's not as regular as we saw with the 200 base pairs in the histones. It's actually a much more irregular looping structure where they attach periodically to this protein scaffold. That protein scaffold can then be further condensed depending on if it's transcriptionally active or not. This looped structure attached to a protein scaffold can be condensed even more if it's what we call heterochromatin. Heterochromatin means that it's transcriptionally inactive. It's not undergoing transcription. Therefore, it can be very tightly packed, or it can be loosely packed. You don't see this tight structure if it's going through transcription. It'll be a much looser structure. We call it euchromatin if it's transcriptionally active. Now, I think we have a picture here. Yes, I have a picture to show you. Inside of the cell, you can actually see the difference between heterochromatin and euchromatin. Looking inside of the nucleolus is where we are, because that's where any transcription would be occurring. What you're seeing is heterochromatin is tightly packaged, not undergoing transcription. Euchromatin is undergoing transcription. It's much more loosely packaged. This is during most of the cell cycle. You would see a structure like this, where some is very condensed, it's not being used. Some is much looser, it is being used. Of course, during replication, it has to be further condensed because you have to have actual lining up of chromosomes during mitosis, or meiosis for that matter. So this structure gets even more condensed when we start to move into mitosis. And this structure we think of with DNA chromosomes, we think about this kind of long X structure that really only exists during mitosis. DNA is not so tightly condensed during the rest of the cell cycle. So they're condensed into these chromosome structures only before DNA replication. Before mitosis, before the nucleus divides. They would not be that condensed at other points.
So if you were to look at, say, a skin cell versus liver cell, you find then the heterochromatic portions
would be different.
So
You can't see it by eye, but yes, that would be true.
But so
Yeah.
it's a way of just hiding it so that it doesn't become activated in a particular cell.
Well, what will happen is the cell, as it differentiates, it will start to express only certain proteins and not express other proteins.
So
And that's
the
what
ones
makes
that
them
aren't
actually
being
different.
expressed are just condensed and put in the
Right,
cell.
right. They're not being used, but the DNA exists there, but they're not being expressed.
What causes them to be condensed and
during the cell cycle, there'll be signals that are sent that causes that condensing of the DNA. There's actually several steps that have to happen for the cell to go through replication. Some of them are internal signals and some are external signals. Actually, some of them we'll talk about today. But so you have to have, most of them are signal transduction pathways. Something comes in from the outside, binds to a receptor, which eventually sends signals down, and you get creation of proteins that assist in the condension, or you'll start to get proteins that are assisting in the breakdown of the nuclear membrane to allow the splitting of the nucleus during mitosis. So usually this is all signal transduction that eventually results in that change. Sometimes life is just amazing. That's what when I was first learning cellular structure, I remember just being blown away by what can happen in one single cell.
Why the X structure?
Why the X structure? You mean from an evolutionary perspective, why is this beneficial versus another structure? I don't know that I've ever heard an answer for why this would be beneficial versus another. And the X structure is certainly very different in different chromosomes because the location of the centromere changes dramatically. So I can't even put forth that having a true X is beneficial over having, kind of that elongated Y or others. I don't know the answer to that. I don't think I've ever come across that. Why an X versus something else? It's a good question. I don't know. All right. So all of the levels here of condensing the DNA into this structure. Here we have the DNA wrapped around the histones to form nucleosomes. Then another histone protein attaches nucleosomes together to form that 30 nanometer fiber. Those get looped onto a scaffolding structure. That protein scaffolding can be even more condensed to form heterochromatin versus euchromatin, depending on if it's transcriptionally active or not. And of course, only during mitosis it gets condensed down into the structure we recognize, this chromosome structure. This is actually a picture. This is one of my favorites. What they've done is they've taken this chromosome and they've degraded only histones. So the actual scaffolding protein is there. You can see the scaffolding protein here. And the DNA has now started to release because the histones are no longer holding them together. All of this DNA is attached to this tiny little protein structure through histones. So it's actually a really amazing packaging ability to get all of that DNA into one tiny little area. All right. Questions on DNA packaging?
I just was curious as to the part D of that. Would that be considered a quaternary structure?
They don't usually use those terms because this is not nearly as predictable a structure. It seems to be more random depending on the cell that you're talking about. So they don't usually use that, but you're talking about an amino acid structure, primary, secondary, tertiary, quaternary. This you always see to this level. From here, things get much more varied depending on what's happening in that cell at that time.
So the previous structure, would that be considered tertiary at that point?
I've never heard those terms associated with this. I suppose the reason is because when you're talking about primary, secondary, tertiary, quaternary in a protein, it's all the amino acids. We're not adding in another biomolecule, and here we're talking about both DNA and protein, so I don't know if that term fits. I've never heard those terms used with DNA structure packaging. All right, let's talk about some more specifics on a cancer cell versus a normal cell. Last time I talked a little bit about kind of the basics on cancer and opening to terms that we use commonly when we're talking about cancer cells. What I want to talk about today is talk about how cancer cells are different from normal cells. Well, we're going to talk about the whole rest of the semester, but we're going to start with kind of basics on how a cancer cell is different from a normal cell and how you could describe it, what kind of profile it has. So some distinctive traits of cancer cells versus normal cells. The first, of course, that they form tumors. Remember from last time, tumors are these uncontrolled growth and cell division, a mass of cells that's undergoing cell division. Remember that in a normal cell, cell division is associated with cell differentiation. So as the cell becomes more differentiated, it is not able to undergo cell division. Right, from last time we talked about that. So as a cell has a clear path in life, it becomes differentiated, it does not retain the ability to go through cell division. But in tumors, that's not true. You can have these differentiated cells that are undergoing quick cell division. Unfortunately, the way they test this is they take a cell and they inject it into the appropriate organism and look to see if it forms a tumor. That's how they decide if it is capable or not. Now for humans, we don't do that in humans. We inject them into nude mice, mice that don't have an immune system to recognize human proteins, so it won't attack it. The next one we're going to talk about some more, but to introduce it, they have reduced sensitivity to density-dependent inhibition of growth. I'm going to go through that in detail in just a moment, but what that means is normal cells will not overgrow in a population. Cancer cells don't care about that. They don't care about cell density. They will keep undergoing growth regardless of what's happening in the environment. Cancer cells are anchorage independent. I'm going to show you examples of this as well. Normal cells have to be attached to something for them to grow. If they are not attached to something, they will die. They will actually go through a very careful process to kill off cells that will not attach. So cancer cells don't seem to have that problem. They will be able to grow even when they are not attached to something. I'll show you examples of that as well. And cancer cells are immortal. They can continue to divide repetitively, regardless of any other outside forces. They can continue to divide. Normal cells, of course, cannot. You already know a big reason for that is because of the telomeres, right? Normal cells will be limited in their numbers of cell division by telomeres. Eventually, the end of the chromosome is degraded. They can't go through cell division any longer. Cancer cells get around that. One way is by turning on telomerase, and so they can continue to go through cell division. There's all sorts of examples. In fact, the book that they're reading on campus, I don't know if you guys know that kind of stuff because you're science students, but you know they have this One Book, One Campus program every year where they choose a book. This time they're using The Immortal Life of Henrietta Lacks, which I actually read last summer. It's fascinating. It's a really fascinating book. I lent it to my friend and never brought it back, so I had to hunt it down. But those cells, the HeLa cells, are a cancer cell line that was obtained from a woman's uterine cancer tumor. And in fact, those cells, which were obtained in the 50s, 54 or something like that, they were obtained in the 50s, are still growing in culture today. And the reason is telomerase. They have the ability to extend their telomeres. That same cell line is still growing today. Let's go through some more of this in more details. Density-dependent inhibition of growth. So growth patterns. I went through how to present this the best, and I thought, you know, the easiest way is to say, okay, what does the normal cell do, and then what does the cancer cell do? So I'm going to use this pattern over and over again, where in blue I talk about what normal cells do versus in pink what the cancer cells do. So normal cells will divide to cover a surface in a monolayer of cells and then stop. Normal cells are extremely responsive to density. They will not overgrow their media. They will not overgrow the container that they're in. They are extremely sensitive to density. So if you put a couple of normal cells into a flask, the cells will actually grow to cover the bottom of the flask in a monolayer. They will not grow beyond that. Right? They don't like to be too crowded. Versus in a cancer cell, that trait is called density-dependent inhibition of growth. The cell density themselves will actually inhibit growth. The only way to keep your cell line growing, if you work in a lab where you're culturing eukaryotic cells, the only way to keep your cell line growing is to go in and scrape off some of these cells, put in new media, and then they'll continue to grow. But you have to go in and scrape them off periodically because you have to give them space to grow, otherwise they'll stop all by themselves. Cancer cells, on the other hand, do not stop at the monolayer stage. They will pile up. They will continue to grow. They're not sensitive to density signals. They don't care about density signals, completely independent of that. So they show density-independent growth, where they will pile up and continue to divide regardless of the density signals that may be being sent. It's not surprising to know if we were to take cells from this area, right, a monolayer of normal cells, and we were to inject these into an animal, is it likely to form a tumor? No. But if we were to inject those cells, is it likely to form a tumor? Absolutely. And in fact, the experiment has been done here. Cells that are less susceptible to density-dependent inhibition of growth, let you work through that. Cells that are less sensitive to density-dependent inhibition of growth, the cells that pile up, they don't care about density, are much more likely to form tumors rapidly when injected to mice. So what they're looking at here is the cell density at which growth stops. Okay? So you take one cell, put it into a culture media, allow it to grow. The question is, when does that massive cell stop growing? Well, here it stopped growing relatively small density. Here it took a long time before it started growing. So this is the really dangerous ones that are no longer responding to density-dependent inhibition of growth. Take one of those cells from that mass, inject it into an animal, and look at how long it takes it to form a tumor. And what you see is those that are most dense, those that care the least about density signals, form tumors very, very quickly. Those that stop early, right, more normal cells, they're responding to the density signals, take much longer to form tumors or never form tumors. So it's not really surprising to know that those cells that are no longer responding to density inhibition are also those that are going to form tumors faster and more aggressively. The next one here, we talked about attachment briefly. Turns out that normal cells are what we call anchorage dependent. They have to be attached to something. Normal mammalian cells are anchorage dependent. They have to be attached to something. Normal cells only grow when attached to a surface or in the extracellular matrix. If they're in the body, they have to attach to that extracellular matrix. They use proteins called integrins to attach themselves to a surface or the extracellular matrix. Normal cells must be attached. Right? If they're not attached, they will actually die through a cell death process called apoptosis. If they can't attach, normal cells will undergo a type of apoptosis, which is programmed cell death, called anoikis, which prevents cells from floating around. The whole idea here is you don't want a liver cell to get detached and float around to some wrong location and start growing liver cells in the wrong place. So if a liver cell becomes detached from the liver, it'll die all by itself through this process of anoikis, which is programmed cell death. Have you guys learned about apoptosis before? Yeah. All right, so programmed cell death, this will happen to prevent them from floating around. Cancer cells are anchorage independent. They don't feel the need to be attached to anything. They will not die off through apoptosis when they are released from a solid surface. They grow on that solid surface or in a liquid. They don't care. It doesn't matter to them if they are attached or not. A little diagram here showing this. Normal cells versus cancer cells. Normal cells, of course, if you allow them to settle, they will attach to the bottom of the flask. They'll grow in their monolayer. They'll be very happy because they're anchorage dependent and they're also responding to density signals. Here, if you keep that flask moving, right, they're never able to attach to the bottom. They will not grow very well. They won't grow if they're not attached to a solid surface. Cancer cells, on the other hand, cancer cells, if they attach to this bottom, if you allow that flask to settle, they'll attach to the bottom, they'll grow, but they won't stop at the monolayer phase because they're not density dependent. And they will also grow into this great big pile regardless of attachment. Same is true if you keep that flask shaking. If you were to keep the flask shaking, they will still grow. They will be not only not recognizing density signals, they also won't care about attachment. They will be able to grow anchorage independent and density independent.
So apoptosis isn't an immune response, it's actually something that the cell does.
Apoptosis can be brought on by an immune response, but it's also part of normal developmental pathways. I'm going to talk about apoptosis in probably towards the end, but it is a normal process for cells to undergo. All cells have the ability to undergo apoptosis if they get the right signal. And one signal can be like a viral infection for immune responses, but it's part of normal development. For example, apoptosis forms our fingers, which, by the way, the baby already was doing this, right, which is crazy, 18 weeks. But yeah, so what happens is we have skin between our fingers, finger webbing, right? What happens is apoptosis kills all of that off during development. So it is normal part of the developmental cycle. There's nerve cells that undergo apoptosis early in childhood, which causes interactions that allow better memory. So, anyway, so another growth pattern here. Normal cells grown in culture will only divide a limited number of times before they degenerate and often die. A lot of work was done with fibroblasts on this particular topic. Turns out fibroblasts will divide about 50 to 60 times before they completely degenerate and can no longer undergo appropriate cell division and no longer even look like fibroblasts any longer. And that's what we're looking at here. This is normal fibroblasts put into cell culture. If these cells are allowed to undergo cell division 50 times, they look like this. Right? Hardly even recognizable as the same cell. You see changes to the nucleus, which are dramatic. You can hardly even see the boundaries around the nucleus any longer. These are the older cells after having undergone so many rounds of cell division. Now, exactly. So what limits the ability for them to continuously divide? Partially, large portion is really the telomeres. Telomeres are shortening. As the telomeres shorten, you have chromosomal damage. Cells are no longer able to divide properly. They will often degenerate and die by themselves. Normal cells will. Cancer cells, however, will continue to divide indefinitely and appear to be immortal. And of course, they're doing this because of telomerase, that enzyme that they turn on that allows them to extend their telomeres.
I'm just curious, because you say that over a period of 50 cells, they basically
50 rounds of cell division.
50 rounds of cell division, they die off. So technically, we'd all be dead by now if there wasn't some other process.
Our cells actually don't undergo that many rounds of cell division. Well, we're talking about differentiated cells, right? The undifferentiated cells can still undergo cell division. It's the differentiated cells that can't undergo cell division for very long. They cannot maintain it for very long. So in an adult, we do retain some cells that have telomerase active that are able to produce, new cells, the stem cells. But as you get more differentiated, they can't do that any longer. And if they do undergo rounds of cell division, they will eventually die off from the chromosome damage.
So I guess so for skin cells, those would be differentiated?
Yes. On the surface of our skin, they're differentiated. Much lower in the tissue, they're undifferentiated, and they retain the ability to divide. And so what happens is each time it divides, it releases one that's more differentiated and not able to divide, and one that is still able to divide and undifferentiated. So you can constantly make new ones from that group of cells that are at the bottom. Cells on the top are done. Those are highly differentiated, but the cells underneath retain that ability.
So they're creating like a young cell and then an older cell?
Yeah. Yeah. So what happens is, remember, differentiation and cell division are coupled together. So as it becomes more differentiated, it cannot go through cell division as well. The reason is because of this phenomenon and the shortening of the chromosome. So it can't go through so many rounds of cell division. So on the telomeres here again, this is showing you that the telomere sequence protects for several rounds of cell division. The telomere gets shorter. Eventually what will happen is you'll have chromosomal damage. And you work through that telomere sequence. It's getting shorter and shorter and shorter. You start to work into the areas of the chromosome that do code for various different proteins. The other thing that happens is, I mentioned this, but we didn't see a picture of it. Remember I told you first off that the telomere does loop around and connect in. That protects against degradation of the end of the telomere. And I was telling you there were proteins that helped that. Here's a picture. They're showing you these telomere capping proteins that actually hold the telomere in that curve to prevent degradation. What happens is, as the telomere gets shorter and shorter and shorter, the proteins can no longer attach. They no longer have space to attach to the telomere. And in fact, the telomere can no longer loop in and form that loop structure. So even here, we're going to have problems. There is still telomere sequence here, but it's extraordinarily short, which means it can't loop around to protect the end of the chromosome. You'll start to get degradation because of the exposure to the aqueous world. You'll start to get degradation of the end, not just because of shortening of the telomere, but in fact because it can't loop around and protect itself any longer from that degradation process. So it's kind of a multi-problem here. Telomere is getting shorter. Eventually you could degrade a gene if it gets too short. It's so short that it can no longer retain the proteins that protect it against that degradation. So both of those are going to cause problems to the telomeres. I'll make sure Interesting thing to note here, normal cells will actually trigger apoptosis if they lose their telomere capping proteins. That's how important they are. So if a normal cell senses it has lost the ability to attach to those telomere capping proteins, that will actually trigger apoptosis. This is probably part of how cells lose the ability to continue dividing. So loss of telomere capping proteins will trigger apoptosis. In a normal cell. To prevent degradation to the chromosome and possible mutation being passed on. A lot of notes, but I think I said it all. Oh, another interesting thing to note. We know about telomerase. Cancer cells will sometimes turn on telomerase to extend those telomeres. Question? How do telomeres get shortened? Remember, with each round of cell division, right, it happens naturally. So as each round of cell division happens, because of the phenomenon of DNA polymerase only extending in one direction, you wind up with a shorter chromosome each time. Just normal cell division will cause that to happen.
What about other things in the cytoplasm that aid in degradation of the telomerase?
Well, there's of the telomeres. The telomeres, well, it will happen to any piece of DNA. It's not specific to the telomeres, but because you have this DNA kind of hanging off into the aqueous environment, what will happen is you'll eventually get degradation of nucleotides just because they're hanging out there.
It's kind of like the AAs on the RNA?
Yes, yes. So that's why it likes to loop it around. It protects the ends so that the ends aren't available for that degradation of the end nucleotides. And then the proteins help it to loop around like that. So cancer cells, a couple of ways cancer cells can get around this phenomenon. First, of course, is by turning on telomerase to extend the telomeres. Cancer cells can turn on telomerase. And that is the most common way to gain immortality, is by turning on the telomerase enzyme. The other thing that's been interesting that they're just starting to recognize is some cancer cells develop a mechanism where they can copy a telomere sequence from one chromosome to another. So what they'll do is they'll actually attach the end of one chromosome to another and copy part of that telomere sequence. So it's only beginning to understand how they do that, but a mechanism to copy a telomere sequence between chromosomes. current thought is they're doing that by actually attaching the end of two chromosomes together. The telomeres actually attach together through base pair rules, and it allows some copying to occur that extends the telomere on both chromosomes. So possibly by attaching chromosomes together at the telomere. That's not nearly as common, I think, as telomerase. Telomerase is definitely what we see in many immortal cancer cells. But there's also this one that's been recently identified where telomeres of two neighboring chromosomes can possibly attach to one another and allow replication to extend the telomere on one of them, allowing some kind of a mechanism of extension through a whole new thing that we haven't seen before. So interesting possibility there. All right. The next one I want to address here are growth patterns. Growth patterns relating especially to cell division, which is dramatically different between normal cells and cancer cells. This is really abnormalities in signaling systems. So how does the signal system differ between a normal cell and a cancer cell? Normal cells will only divide when they receive the appropriate protein growth factor. So a normal cell cannot go through cell division unless it gets a protein we call a growth factor. Absolutely required for that cell to go through division. We're going to see the process of how that's controlled in just a moment. But normal cells can't go through cell division without that growth factor. Two very common ones that we see used in human cells: platelet-derived growth factor, which comes from platelets, that's why they called it that. Right? Platelet-derived growth factor is involved in connective tissue and smooth muscle cell division. Epidermal growth factor seems to be involved in all sorts of different tissues in their cell division. It's very widely used by lots of different cells. In all cases, growth factors are ligands that attach to a signal transduction receptor, just like we've seen before. So here's epidermal growth factor attaching to the receptor, signals through a signal cascade that eventually causes the production of some protein involved in cell division. So it's causing a change in expression. Normal cells are absolutely reliant on these growth factors. In fact, we're going to see the cell cycle in just a moment. They pause and they wait to get their growth factor. They will not go through cell division. They cannot go through cell division until they get their growth factor. This is one of the regulation steps in cell division, is to require a growth factor.
Are these growth factors found in plasma mostly?
Yes, mostly. Well, mostly they're circulating through the bloodstream and the lymph. So they would be circulating through the whole body.
Because I was reading another article where they started, the FDA's come down on it, but they're taking blood from a patient, for example,
Right.
infusing it, and then injecting the plasma back in, because it's supposed to be rich in growth factors and, I forgot what the other thing is called, T cells.
Okay.
And then they inject it back into areas which have low circulation, and it's supposed to infuse that area and promote.
Growth factors will promote division, especially if we're talking about like platelet-derived growth factor would help in formation of new blood vessels into the area and extension of capillary beds. So that does make sense if it's low circulation. I thought we were talking about a cancer treatment. I was mystified.
No, not a cancer treatment, but
Yeah.
I just was curious where it was located.
Platelets, yeah, it is circulated through the bloodstream, and platelet-derived growth factor is also involved in extension of capillaries. So you would get additional blood flow to an area if you could stimulate more release of platelet-derived growth factor. In fact, it will trigger the production of capillary beds where they don't already exist. It's actually a really powerful growth factor that way. So in both cases here, it's a signal transduction process that's triggered by those growth factors. Growth factors stimulate tissue growth during development. They also are active in wound repair. So during some kind of a wound, you could get production of growth factors to stimulate new tissue being created. You also see it present in small quantities in adults to replace old cells. So old cells are replaced through the release of growth factors, allowing the cells to go through their cell division. Now, cancer cells, on the other hand, divide independent of a growth factor. They do not need that growth factor to go through their cell cycle. Now, if you had to suppose, how could a cancer cell get around this issue? How could a cancer cell possibly become independent of external growth factors? Any hypotheses?
Be a problem in the ligand,
Problem in the ligand?
where it just automatically conforms so that it goes through the signaling pathway to.
Say that. So you're thinking the ligand is somehow
Or, I'm
different?
sorry, not the ligand, the receptor.
The receptor?
Yeah.
All right, do you guys agree that the receptor could be? Sure, absolutely. The receptor could be mutated. If the receptor is mutated, you would want a mutation that causes automatic signaling without the need of a ligand, right? So you would want it to constantly signal and be sort of ligand independent.
So what if it was like a core and that was like this open?
One more time.
Never mind.
That's okay. There's no. You may come up with one that we don't know of yet, too. So receptor mutation, so it's ligand independent to constantly signal. What about if that cancer cell had the ability to produce something all by itself, so it didn't have to wait for it?
It produces its own ligand.
Its own ligand, its own growth factor. Some cancer cells will make the growth factor themselves. Very clever. Or they'll make a hyperactive version of the growth factor. So they'll make a really super active version of that growth factor. Make their own growth factor. Right? Or kind of a hyperactive version of it. All right, where else in this cascade could we exert some change? We've talked about what we can do with the ligand, talked about what could happen to the receptor. What else?
They could the reinforcement in the protein.
Over-amplification?
Over-amplification, so you're talking about where in this process?
I don't know exactly. One of those little shapes.
Sure, one of these, remember how a cascade works, right? You have your ligand binding to a receptor. Receptor activates a couple of the next guys. Those guys activate a couple of the next guys to get amplification of response, which is what you're talking about. So it's not just the receptor that could be mutated. It could be any of these proteins in that cascade that could be mutated to be hyperactive, to cause an excessive response. So it could also not be a receptor mutation, but a hyperactive version of some of these signal transduction proteins, somewhere in the pathway. So hyperactive version of a signaling protein or proteins. And those are the three that we have identified so far as to how they can become independent of this signal. Either they mutate the receptor. If they mutate that receptor, the receptor has to become growth factor independent. So it's constantly signaling, constantly saying go through cell division. Or it could make its own growth factor. Generally, that's a process where it's just self-signaling constantly. It doesn't need to wait for an external source of the growth factor. Or it could mutate some part of this signal transduction cascade. Anywhere in there, if it mutates those proteins to become hyperactive, you have constant signaling. All of those are possible ways for a cancer cell to get around the need for outside signaling. In all cases, this leads to either all of these All of these would lead to either overactive signaling, overactive signaling if it was making its own growth factor, that would mean it was overactive, too many rounds of cell division, or if it was this hyperactive version of a signaling protein, that would be overactive. Or it would be totally autonomous. It doesn't even care about growth factor. If it had its receptor mutated, it would be totally autonomous and not worry about signaling from a growth factor. Or totally independent. Which essentially means it's going through cell division much faster than it should. It's not listening to the normal system that's set up through growth factors in this signaling cascade.
Like intracellular signaling, non-infected cells, like cell division.
So how would intracellular signaling in a normal cell, say it one more time.
Yeah, with an infected cell.
Infected or you mean cancerous?
Yeah, cancerous.
So signaling of a cancerous cell versus a normal cell? So if we had two cells, well, a cell would have to be one or the other, right? So here, what I think I'm going to answer your question. If not, ask it again. So a normal cell, you'd have ligand bind to the receptor, signal to the first protein, signal to the second protein, and eventually cause some change in cell expression. Remember ETS and June that can cross the membrane, cause a change in cell expression. But in a cancer cell, what we're saying is it's possible that maybe the orange one has been mutated so that it doesn't just signal once the way it's supposed to the red guy, but instead it's signaling 10,000 times. So that one little ligand binding will cause many rounds of cell division, making it much more sensitive than it should be to that growth factor if it was cancerous. That's one way it could be different in the actual signaling cascade versus a normal cell where it shouldn't go through that kind of process. Did I answer your question?
What if the infected cell tells all the other cells not to divide?
What if the cancerous cell told the other cells not to divide?
Yeah.
I don't know if it would do that because in its perfect world it would want everybody to divide.
Something like the embryos, the intracellular signaling.
Right, in a normal cell, it will send out signals that will, so in an embryo during cell division, right, it will send out the appropriate growth factor to trigger cell division, or we will see, actually maybe this is what you're asking about, we will see it's possible to send out a death ligand as well that triggers apoptosis. Actually, I think, is that going to answer your question about it? All right, it is possible to send out a death ligand that will trigger apoptosis if you want a cell to die. That happens during some viral infections where one cell is infected by a virus and it will send out signals to neighboring cells that will actually kill off cells in the area in order to avoid viral spread to the tissue. Or if it's time for that cell to divide, like in the finger webbing example, it will send out death signals that cause apoptosis. All right, so ways that the cancer cell can become independent of these growth factors. So let's talk about this cell cycle and where this would occur. So the cell cycle, you've learned cell cycle before, yes? Right, so G1, S, G2, M phases here. G1 is a growth phase. Length of time varies dramatically. On an average for human cells, it's like 8 to 10 hours. So is the G1 phase. S phase is DNA synthesis phase. This is where DNA replication is occurring during this S phase. It takes about 6 to 8 hours for that to happen in a human cell. G2 phase. G2 phase is a second growth phase. Really, that's where the cell is preparing to go through cell division during this G2 growth phase. And then M, of course, stands for mitosis, although cytokinesis happens during that time frame as well. So the M phase includes both mitosis, separation of the nucleus, and cytokinesis, separation of the cytoplasm. length of time for a cell to go through the whole entire cycle is extremely different depending on the cell that we're talking about, the stage in development that we're talking about. The quickest that it can go through this entire process is about, it's 18 to 24 hours is the fastest it can possibly happen. So the fastest, like a cancer cell that's not waiting for signals, the fastest it can go through cell division is about 18 to 24 hours. Now, this cell cycle, right, with these different phases is extremely regulated, highly regulated process that the cell goes through to follow this cycle. Now, regulation of this cell cycle, why is it so important? First, you monitor timing. Right? You do not want cells to go through cell division when they're not supposed to, so you can monitor timing on a tissue level, a developmental level. Also, it makes sure that each phase is completed before the next phase starts. For example, you need to make sure that the DNA replication has completely finished during S phase before you start the next phase. It's important to make sure that one phase is totally finished before the next one starts. Another reason to regulate the cell cycle so closely. This cell cycle responds to both external and internal signals. It will respond to signals from the outside environment as well as signals that are internal as well. Mm-hmm.
How does mitochondrial replication, how does it line up with the full cell?
Mitochondria actually will go through cell mitochondrial division without needing to follow the whole cell cycle. Mitochondrial division can happen in G1 or G2. Very rarely during S, actually. Usually the cell is so devoted to what's happening in the nucleus, there's not enough ATP to drive the process during S, but it can happen in G1 and G2, and you don't have to have a certain number of mitochondria to go through cell division.
So, cancer cells still needs them, right? So
cancer cells still needs them.
Does it somehow boost the speed that its mitochondria replicate, or?
I don't know of how it does this, but it just intuitively it seems like it's got to. It's got to be causing some signal release that would cause mitochondrial cell division, because the cancer cell is going to need additional ATP to go through so many rounds of cell division. I don't know the signaling pathway, but just intrinsically it's got to be doing that. I totally agree. Because it's got to have a lot of ATP to drive all of the processes that are happening in that cancerous cell. Although cancer cells don't stop, no cell thinks, but cancer cells don't stop to think about anything. They just keep going. But just the fact that so much ATP is needed to drive these processes, it must be getting additional signaling to the mitochondria. So let's talk about a little bit more about this cell cycle, what's happening along the way here. We just walked through it, but G1, we're talking about the first growth phase. They're showing you in green the amount of DNA per cell. They're showing you in red DNA synthesis activity. During G1, it's mainly a growth phase. That's when the cell is doing its normal business, producing its normal proteins, going through whatever metabolic processes are normal for that cell during the G1 phase. During the S phase, it's going through DNA synthesis. So you're seeing lots of DNA synthesis activity here. You're also seeing an increase in the DNA level per cell. Right? You now have 2 copies of the chromosome. G2, the cell is preparing for cell division, producing whatever proteins are needed to finish that cell division process. And in mitosis, the cell divides, so the DNA per cell goes down as you get back to normal levels for each cell. You've also heard the term interphase. Interphase is everything except for mitosis. So that includes all of that includes G1, S, and G2 would be interphase for that particular term. Now there is a control point that becomes really important to the cell. Here they're showing you what's called the restriction point. The restriction point is what's going to stop the cell from entering into S phase. This is what really controls whether or not the cell is going to divide. Because once it enters into S phase, it's going to go through the rest of the process. So So this is sort of the stop signal for, is this cell going to stay in G1 or is it going to move into S phase? That's called the restriction point. The restriction point, what do you suppose needs to happen to push the cell into S phase? You need a growth factor. Binding of a growth factor to the receptor is what's needed to push a cell through its restriction point. It has to have that growth factor. If the cell gets its growth factor, it pushes through the restriction point, it goes into S phase and eventually goes through cell division. If the cell never gets its growth factor, it enters into what they call G0, which is kind of this eternal wait for a growth factor. It's normal cell activity, but it's this pausing point. It's waiting for its growth factor. It cannot go on without that growth factor. Interestingly enough, a cell that's been in G0 for a long time frame, if you expose it to a growth factor, it will push it on through that restriction point. So it is literally just waiting for that growth factor to show up. All right, let's talk about how a cell can control moving through something like a restriction point and other points in the cycle. We talked about growth factors, and I'm going to get into the nitty-gritty of how a growth factor affects that in just a moment, but I want to talk about protein that's involved, and then we'll kind of put the pieces together. Is P53 on here?
No, but like in general.
Okay. I was going to say, did I have it that early? We're going to talk about P53 in a few minutes. It's really important for pushing the cell through the cell cycle, and it's a protein that's involved in that process. And actually, I've got several slides on them that are coming. So I'm going to put that on pause because I'm going to get there. And in fact, P53 is so important that half of all human cancers are some kind of mutation to P53. It's really important in the cell cycle. All right, so moving through the cell cycle, what controls movement along the way? What controls which point in the cell cycle the cell is undergoing? I said that kind of weird. But where in the cell cycle the cell is depends on really two different proteins, cyclins and cyclin-dependent kinases. So cyclins and CDKs, with a name like kinase, you already know that it's going to phosphorylate. Kinases by phosphorylate. So cyclin-dependent kinases, once they're active, will phosphorylate. And they are called cyclin-dependent kinases because they depend on another protein called cyclin. Right, so the CDK, the cyclin-dependent kinase, has to be attached to his cyclin in order to do his phosphorylation. So CDK attached to his cyclin is going to cause phosphorylation of proteins, and that will trigger a movement in the cell cycle. Now it turns out that there are several different cyclins and several different cyclin-dependent kinases because they're needed for different points of the cell cycle. So there's ones that are involved here. They're showing you the transition between G2 and M. There's a handful of cyclins and cyclin-dependent kinases that are used to push through that point. There's another point that requires them in another part of the cell cycle. So cell cycle. Cyclin and cyclin-dependent kinases are named for their phase change. For example, there are the mitotic CDKs and cyclins. Those are the ones that are showing you in this one, in this diagram. So the mitotic CDKs and cyclins are needed to enter into mitosis, to go from G2 into mitosis. There are also cyclins and CDKs that run here between G1 and S. The restriction point that we saw a few minutes ago, part of that process is actually run by cyclins and cyclin-dependent kinases as well that are involved in the transition from G1 into S at that restriction point. Now the background here. Cyclin-dependent kinase levels remain constant in the cell at all times. So we always will have levels of your CDK, right? CDK, cyclin-dependent kinases, are pretty much always in the cell, and they're around. but they're not active because they're waiting for their buddy, the cyclin. Right? They cannot be active until they get their cyclin. So CDK levels remain constant throughout the cell cycle, relatively high levels constantly throughout the cell cycle. What will happen is the cyclin level changes. Okay, so CDK is constant. Cyclin levels change. CDK levels are constant, but the cyclin levels change through the cell cycle. When the cyclin levels are high enough, it's going to bind to its CDK and activate the process. So high cyclin allows binding to the CDK and activation of this whole process. You guys with me? So CDK levels are constant, cyclin levels change. When the cyclin levels are high enough, it'll bind to the CDK and allow the process to continue. And this will push it through different phases of the cell cycle. All right, so let's look at this particular example. We have constant levels of our CDK. He's inactive because he has to have a cyclin to attach to, but he's at relatively constant levels throughout the entire cell cycle. Cyclin is being produced at low levels. Eventually those levels increase. When the cyclin levels get high enough, cyclin and CDK bind together. Right? That's the first step of activation of cyclin and CDK. Once cyclin and CDK are bound together, cyclin-dependent kinase is now active, and a kinase is going to phosphorylate. What does he phosphorylate? It turns out that kinase phosphorylates a whole bunch of proteins that are needed for mitosis. He will phosphorylate proteins that break down the nuclear envelope. He will phosphorylate proteins that cause condensation of the chromosome. Remember the chromosome coming together into those individual chromosomes. He will phosphorylate proteins that start the formation of the spindle needed in mitosis. And various other proteins get degraded that are inhibiting the process. So what I'm trying to get to here is when cyclin and CDK come together, they phosphorylate proteins needed for mitosis. They activate the mitosis process. Mitosis can't happen if these two proteins aren't bound together.
Is there a name for that complex of the mitotic cyclin?
No. There's not. I'm not unfamiliar with any name for that. That's complex together. It's an active CDK. CDK is not active without his cyclin. So it's active CDK, I guess is the right term. All right. This is the particular process that happens in the entry into mitosis. Turns out that there is a protein produced by one of these. So one of these actually causes the production of a protein that will degrade the cyclin and allow the process to start over again. This is important because if you have long-term levels of both cyclin and CDK together, you're going to push the cell through multiple rounds of mitosis. That's not good because you have to wait for DNA synthesis before the next round of mitosis. So you have to control it tightly. So what happens is they get together, the CDK phosphorylates a number of proteins, and one of them that he activates is actually going to cause the death to his partner, cyclin, which is important. If you don't do that, you can't stop the process. So binds to CDK, CDK phosphorylates proteins needed for mitosis. Interestingly enough, he also phosphorylates and activates this anaphase promoting complex, which will degrade the cyclin. And so CDK phosphorylates proteins needed for mitosis and degradation of the cyclin. CDK CDK phosphorylates proteins needed for mitosis and degradation of the cyclin, so that you have one round of mitosis and no more than that with each round, each time the cell goes through that cell cycle process. You guys okay with this, or should you want me to do it one more time? I feel like it's a little tricky. Yes, we're good, or one more time, please? All right. I'm hearing both. I'll do one more time, but I'll do it quickly. All right, so we have we have CDK levels pretty constantly, pretty constant levels in the cell during the entire cell cycle. This mitotic CDK is at pretty constant levels. What changes is the production of cyclin. We have almost no cyclin present at the end of mitosis. Once mitosis is over, cyclin levels begin to increase. More of that cyclin protein is made. Eventually, the levels get high enough that mitotic cyclin binds to mitotic CDK. Now CDK is active. He begins to phosphorylate those proteins that are needed for mitosis to occur. He also phosphorylates a protein that will degrade mitotic cyclin to block multiple rounds of mitosis and allow the cell cycle to continue. This particular point that we're talking about is the entry into mitosis. So what triggers the entry into mitosis is these mitotic cyclins and mitotic CDKs binding together to allow that process to occur. Now, it is true that there are other forms of cyclin and CDK that are active at other transitions. This is the one I'm telling you about that's most exciting, but there are other ones at other points in the cell cycle. There is actually another level of regulation to CDK. Some CDKs have to go through their own phosphorylation steps in order to be activated. This exerts another level of control that the cell can have. So regulating CDK activity, of course, we know availability of the appropriate cyclin is necessary. If the cyclin doesn't bind to the CDK, CDK is not active. So here's the first level here of regulation. Turns out that there's additional levels of regulation that have to happen for it to be activated. There's, interestingly enough, a number of different kinases that activate the CDK. And this is fascinating. What happens is there are inhibiting kinases and activating kinases. Now, this is one of the few times in biology where we see phosphorylation can actually inhibit activity. That's pretty uncommon. Usually, phosphorylation activates, but there are some exceptions to that rule. And it turns out that there are some kinases that can phosphorylate CDK and cyclin and cause inhibition. So there are inhibiting kinases as well as activating kinases. And in order for it to actually be completely active, you have to have only the activating kinases there. So the cell has multiple levels of control is what I'm trying to get to. If the cell wants to block replication, if it wants to block mitosis, it can block mitosis by not making the cyclins or degrading the cyclins. Or it can inhibit it by creating these inhibiting kinases. So there's all sorts of levels that can be addressed to block this mitosis process, giving you lots of control over the cell and the cell cycle regulation. All right, so let's put some of this together. We've talked about growth factors. We talked about cyclin and CDK. I want to talk about how these come together in order to push past, remember the restriction point, the point that goes between G1 and S phase here. That is also reliant on CDKs and cyclins. So restriction point control here, putting all of our bits together. First off, we have to have a growth factor. If we don't have a growth factor, game over for a normal cell. Growth factor binds to its receptor, signal transduction. That will go through a signal transduction pathway that eventually activates our cyclin and CDKs. So eventually we have our cyclin and CDKs binding together because of this signal transduction pathway. Usually it's production of the cyclin that changes. So now you have binding of cyclin to CDK. CDK, once he's active with his cyclin, is going to phosphorylate. When we're talking about the restriction point, the protein that's really exciting here is called RB. RB has to be phosphorylated to allow movement past that restriction point. So let's put together all of these. All right, we have growth factor binds to receptor. Growth factor binds to receptor. Receptor will trigger that signal transduction cascade. The signal transduction cascade will produce the cyclin needed. Makes cyclin. What's the cyclin going to do now? Binds to his cyclin-dependent kinase. Cyclin binds to CDK. And CDK is now going to phosphorylate something. And the one we're excited about here is it's going to phosphorylate RB. It's going to phosphorylate this RB protein. That's the basic cascade for getting through the restriction point. So we should clarify this is to pass the restriction point. And the restriction point moves you into S phase. Goes from G1 into S phase. To enter S phase of the cell cycle. So the next logical question is, who's RB and why do we care about RB? RB protein, we're talking about two weird exceptions in biology all on the same day, but the RB protein is actually another one which is active before he's phosphorylated, and phosphorylation deactivates him. So what RB does is the RB protein inhibits the cell cycle at the restriction point. RB inhibits the cell cycle at the restriction point. He actually blocks movement of the cell into that cell cycle and movement into the S phase. When he gets phosphorylated, he can no longer do his job. He's no longer able to inhibit. That restriction point activity. So phosphorylated RB is inactive and can't inhibit the progression. Inhibit entry into S phase. We're going to see that RB is an essential protein in human cancers as well. In fact, most human cancers come down to either a mutation in RB or a mutation in P53. Those are the two biggies when we talk about human cancers. And the reason is because RB is involved in the progression past the restriction point. If you're not moving past the restriction point, you can't go through the cell cycle. So growth factor binds to receptor. Receptor causes the signal transduction cascade to work, the production of cyclin. Cyclin and CDK bind together, phosphorylate RB. RB no longer inhibits the restriction point. You now push past the restriction point and enter into the S phase and move the cell forward into DNA synthesis. Now there are additional guess this is a good place. I wanted to get to P53, actually. Actually, let's do a couple more, and then, yeah, we'll do a couple more. We'll get through these additional steps of cell cycle control, additional checkpoint controls. So we've talked about this DNA, this restriction point that blocks progression. We talked about the one that blocks movement from G2 into M. You know that the group of proteins that are involved in this are the cyclins and the CDKs. Moving it from G2 into M, mitosis doesn't happen until mitotic cyclins and mitotic CDKs are active. There are additional checkpoints as well that are present throughout the cell cycle, and they're maintaining the integrity of the cell cycle. They're making sure that one process has finished before the next process starts. They're making sure that you don't have multiple rounds of any of these processes. So these checkpoints prevent the cell from proceeding through the cell cycle unless the previous phase has already been completed, making sure you don't have doubling up of these processes. There is a DNA replication checkpoint, right? This is the one we've actually seen a moment ago that prevents mitosis from happening unless DNA synthesis is complete. So it monitors DNA replication and makes sure that DNA replication is totally finished before mitosis can start. Part of that is done through the cyclins and the CDKs, and there's other things involved as well. In fact, interestingly enough, if you treat cells with a DNA synthesis inhibitor, so if you inhibit DNA synthesis, if you get them somewhere to this somewhere in this phase and you inhibit DNA synthesis, they will stop at that point, and your cyclins and CDKs will not be active to push it through mitosis. So in other words, they have to finish complete synthesis and replication of DNA before you can enter mitosis. Even if you artificially induce this with a chemical treatment, they still won't go past it because they're waiting for activity by cyclins and CDKs to push it past. There is a spindle checkpoint. The spindle checkpoint actually happens between metaphase and anaphase. Remember, you have to pull apart the two sets of chromosomes. The spindle does that job during mitosis. The spindle checkpoint, you won't move past it unless the spindle is formed properly. And it makes sure that all of the chromosomes are attached to the spindle. Why would you want all of the chromosomes attached to the spindle? So that you make sure you get one copy into each daughter cell. Right? So you won't finish mitosis unless every chromosome is attached to its appropriate spindle, so that when they're pulled apart, they actually wind up with one copy in each daughter cell. So the spindle checkpoint is there to make sure all chromosomes are attached to the spindle. There are also DNA damage checkpoints that will check for damage to the DNA at various different points. DNA damage checkpoints happen in multiple areas of the cell cycle. P53 is involved in this process. We'll talk about how probably next time, actually. But with damaged DNA, P53 will actually trigger apoptosis. So if it senses a large amount of DNA damage, P53 will trigger apoptosis. Why do you want to do that? You don't want to send mutated DNA onto the daughter cells. So P53 is going to kill off that cell through apoptosis in order to prevent it from sending mutated DNA on to the next generation. So cell cycle control, through all of these processes, responds to both external and internal signals. So the external cell cycle control here, if we go back and compare normal to cancer, normal cells only continue through the cell cycle if they are exposed to the correct growth factor to stimulate that signaling pathway, right? So this is the restriction point we're talking about, and it's an external control. Only if you get the right growth factor is it going to go through this restriction point. Cancer cells, through various different mechanisms, can get past that restriction point without worrying about a growth factor. They make excessive amounts of their own growth factor, or they mutate their receptor, or they mutate the transduction signaling proteins. in order to allow them to go through that restriction point, regardless of what's happening with the growth factor. So cancer cells can create a loss of restriction point control through various mechanisms. They don't have to respond to an external signal. For an internal signal, an interesting thing I noted in the literature, if you take a cell, and you grow it in an extremely low level of growth factors, or if you allow it to grow to an extremely high cell density, put down your notes and just listen for a second. This is just interesting. You don't need to worry about them. If cells are growing with extremely low levels of growth factors, in other words, you're depriving them of nutrients. So if you either deprive them of nutrients and their growth factors, or you allow them to get to extremely high cell density, or if you somehow give them inadequate nutrients, they'll stop at that restriction point all by themselves. Okay. Cancer cells will not stop at that restriction point, even if they're starving, even if they haven't finished their cell replication process from the last time. So how are they doing that in starving nutrients? They're making their own growth factor. It's the only way they can get through that restriction point control. But if you take a cell that you have grown for a long time, you've grown it for a very long time in extremely low levels of growth factor, sort of adapted to these extremely low levels of growth factor, you take that cell and you put it into an animal, what do you think the likelihood is of forming a tumor? High. In other words, you sort of force this situation on them where they begin to make their own growth factors. So there's some evidence to show that a normal cell can be manipulated by growing them in these artificial conditions. You can grow it in really low nutrients and low growth factors. You can force it into these situations where they're in really dense environments, like a cancer cell would prefer. You can grow it on media where they're anchorage independent. And if you take any of those and put them into an animal cell, you will get tumor formation at a higher rate. So the environment is controlling these cells, certainly, and causing these mutations to happen, which then lead to problems in the cell cycle. So there's external things that can control the cell cycle. What about internal factors that can control the cell cycle? Normal cells will only continue through the cell cycle if they are healthy and in optimal conditions. Low cell density, they have anchorage, adequate nutrients, etc. That's the only way they're going to go through these internal checks, DNA damage checks, spindle checks, all of these internal checks will only happen if it's in a healthy environment. Cancer cells are not responsive to health signals. They will continue to proliferate regardless of internal conditions and DNA damage. And how do they do that? They found a way to get around the cyclin and CDK process. They found a way to make their own growth factors. They found a way to ignore checking the DNA. That's usually through a P53 mutation. They found a way to avoid caring about the DNA or the spindle. They've mutated ways to get around those checkpoints. So therefore, they'll continue to go through rounds of cell division. So does that mean that a cancerous cell is more or less likely to gain additional mutations? Far higher chances that it will get additional mutations. Once a cell has become cancerous through a handful of mutations, it's able to go through the cell cycle without regulation. What's going to happen is he's no longer caring. He's not paying attention to DNA replication checkpoints. He's not paying attention to the restriction point and spindle checkpoints. So the likelihood of additional mutations is even higher. So their mutation rate goes up dramatically as a cell becomes cancerous, and it's more likely to accumulate additional mutations. This has actually been tested with the thought that maybe we could use this as a cancer treatment. Here's selective destruction of cancerous cells based on loss of restriction point control. Let me walk you through how they did this here. They're adding a couple of different chemicals. Sterosporin halts cells at the restriction point. Well, it's supposed to. Normal cells. So sterosporin point. Probably no surprise to you to know that sterosporin does not work on cancer cells. And they don't care. They don't pay attention to a restriction point. They're going to go through their thing one way or the other. So it only works on normal cells.
Is that along with the RB protein?
Yes, that's the one with the RB, is controlling that restriction point control.
So they both work together to restrict.
Yeah, and so there's the growth factor that's required. You've got to have your cyclin and CDK active. You have to have RB there and functioning correctly in order to control restriction point. Camphoterin. Camphoterin kills any cell that's undergoing DNA synthesis. It destroys cells that are undergoing DNA synthesis. So stops DNA synthesis, which kills cells in that process. Kills cells in the S phase. S phase is where DNA synthesis is occurring. So any cell that's in the S phase is going to be killed by that particular drug, camphoterin. So here's what they did in this study. They took normal cell. They exposed it to sterosporin, which halts cells at the restriction point. So all cells should now be at G1, waiting for that ability to get through the restriction point. They then expose them to camphoterin. That's going to kill off any cell that's in the S phase. But are there any normal cells in the S phase? No. So the cells are fine. If you remove both drugs, the cells are fine. They go back to their normal cell cycle. But for cancerous cells, you expose them to the first drug, which is supposed to block them at the restriction point, but they don't pay attention to the restriction point. So cancerous cells are continuing through that cycle. You expose them to the second drug that kills any cell that's in S phase and undergoing DNA synthesis, and it kills them all. When you remove both drugs, you killed off the cancer cells. This is exciting news. This was possibly the ability to kill off cancer cells without hurting normal cells, because they don't have restriction point control. So when you expose them to the first drug, they don't pause the way they're supposed to. Now, this is not a series of drugs that we can expose the human to in large doses. This works great in a test tube. It works great in a petri dish. Perhaps we could even inject some form of this into a tumor, but we could never talk about giving this intravenously, right? This is not that kind of a drug. But it was the first time that they began to realize we can find drugs that will specifically destroy cells that are going through this uncontrolled process. And maybe not one drug, but a sequence of drugs to kill off those cells that have become cancerous without hurting the normal cells. So this was kind of the beginning of the idea of how do we do that and how do we target just those cells undergoing uncontrolled replication. So selective destruction of cancer cells based on their loss of restriction point control, using that to our benefit in figuring out a new treatment. All right, this is a good place to break. I will pick up here and start the next topic on Tuesday. Let me get this uploading.